Hello, everybody. Welcome to Forensic Science with your instructor, Mr. Antraco. Just trying to add a little bit of flair to these videos. I noticed my viewership is very low, and my subscribers are pretty much my mom. So I'm trying to jazz things up with a little bit more things that are funny as opposed to things that are kind of lame. So my motto is never stop improving, just like Lowe's. So we've got action figures by the yards. We've got some demos that we're going to be doing this week. So hopefully you'll like the extra pizzazz and actually watch some of these videos. So we're on to the next unit. We didn't get to do much in the lines of actually dealing with prints and fingerprints, as we won't get to do much of the blood labs. I'll do these all as demonstrations. But on the positive note, you don't have to do any of the book work, because I didn't have enough books to hand out to all of you before you left, so you don't have to do any of the textbook work. But if we do gather at the end of the year, well, look out. We're going to do some book work so that we get some more of this material and use it as sort of a review. But for now, we're going to kind of do all the fun stuff that you just get to kick back and watch. So I hope you do watch these videos, because they're going to tell you a lot about blood that maybe you just didn't know. Specifically blood type and how blood is used in forensic science. Of course, when you get blood, you also have the almighty tool of DNA in that blood. But the second part of this unit is blood spatter. Notice there's no L there, and that's not a typo. It is blood spatter, not blood splatter. So you can be the actually guy and talk about blood splatter and say, actually it's blood spatter. But anyways, we'll do an activity on that, and you'll get to see how that actually does help very much in the reconstruction. And that's what mostly we use blood for, is recognizing it, analyzing the DNA side of it, and using the spatter marks to try to recreate what happened. So that's the big picture. But I do want you to learn a little bit about blood before we start looking at some of these other videos. I'm going to have a series of videos. This one will walk you through just blood and blood typing, the basics. The second video will talk about the top half of this board, which is what your blood types are. And then the bottom half of this video is a blood typing whodunit. And so I've got all the materials here set up, hoping to have a special guest, one of my sons or both, to be my lab assistants to help us solve the murder of the jewel thief, which we have to look into. But first, let's have the basics. Very simple. I like to look at blood as what's called a matrix, where it is not a suspension, it is not a solution, right? Because there would have to be things dissolved in it, or things suspended in it. And blood actually has both, so they refer to it as a fluid matrix with larger molecules like enzymes and other proteins and hormones. They are suspended in your blood, but you also have gases and amino acids and monosaccharides, things like that, that are dissolved in your blood. So your blood is a solution and a suspension. The most important components of your blood are white blood cells, red blood cells, plasma, and platelets. Of course, when I teach anatomy and phys, and you guys, just so you know, the fancy name for white blood cells are leukocytes. Right? That's where the name leukemia comes from, where you have a whole bunch of leukocytes that don't seem to work very well, an immune problem. Erythrocytes is the fancy name for your red blood cells. That's what we're going to focus on when determining blood type, because it has to do with the antigens on there. But we've also got platelets and plasma. The plasma is the fluid portion of your blood that holds all the nutrients, gases, enzymes, antibodies, minerals. The list goes on and on. this is what your blood would look like if it was spun in a centrifuge, right? Where the bottom half is your red blood cells, because your red blood cells are high in iron, right? Iron is what acts as a magnet to pull oxygen out of the air. The protein that contains iron in your blood is one of my favorite words, hemoglobin. And so that's why your red blood cell would be spun in all the densest part. The iron containing blood would be at the bottom. Then you've got your little section of white blood cells. Notice it doesn't take a lot to keep your blood safe, as well as a giant portion of this is plasma, because of course we've got lots of things. The list goes on and on here of what is in there, not just nutrients that your cells need, but right, this is also where your wastes are dumped into your blood to be filtered out. There's just a funky looking picture of your blood. Now what we're going to focus on is this term down here, serology, right? You have a whole science dedicated to studying these antibody-antigen reactions. Okay? And I will explain more about these other terms in the second video, but I'll give you the brief explanation now, and then we'll cut this off and use the models to explain it better. But just so you know, an antigen is a marker on a cell surface. You learn them in biology class when they were called receptor molecules. Antigens are just name tags on cell surfaces that say, hey, I'm part of this body, Mr. White Blood Cell, don't eat me. All right, where it could also say, oh, I'm a foreign antigen, you should start the immune response and gobble me up. And then, of course, this is where I also sometimes take the chance to talk about allergies, right, where your body responds to antigens that are not harmful so much. So that is the whole antigen, right, something that may cause a response, or it may tell the body that it's part of that. Antibodies are these Y-shaped proteins. Okay, their job is to destroy pathogens or any foreign antigens in your blood. They do that through a process called agglutination. When the correct antibody finds the correct antigen, it will bind to it, called agglutination. Okay, now this is a defense mechanism. This is what's happening in the patients of COVID-19 right now. It's also happening in laboratory offices where they're trying to manufacture a vaccine, where they're trying to construct these antibodies. Our bodies who are exposed to COVID-19 are trying to construct antibodies right now so that they can find those cell surfaces and have the right antibody to knock that out. And so that's what our immune system does. And so this is part immune system, but as we have found out that these antigens and antibodies have different types. And we've gone over the genetics of this and other things in biology class. But I do want to go over this one more time using my models. And so this is what agglutination looks like right here, just so that you can see. When the antibodies connect to these cells that have the antigens, and then we get agglutination, and we can use that information to determine blood type, which we'll do in the next coming videos. But of course, this is just the basics of serology. What we're going to really focus on is blood stain evidence after that. And I've got a whole bunch of stuff that I use for the blood spatter lab that will hopefully help you to understand how scientists use that to reconstruct the post-mortem or whatever, pre-mortem, ante-mortem, I should say, period. So, but that's for a few days from now, probably next week even. So, enjoy these next few videos, and we'll see you.